The overall goal of this procedure is to describe the preparation of Drosophila S2 cells for light microscopy. This is accomplished by first preparing glass bottom culture dishes. The second step of the procedure is to coat the glass bottom with the lectin concanavalin A to promote adhesion and flattening of the S2 cells. The third step of the procedure is to add culture medium and S2 cells to the culture dish. The final step of the procedure is to fix and immunostain the S2 cells in the culture dish. Ultimately, results can be obtained that show localization of fluorophore tagged or immunolabeled proteins of interest through fluorescence microscopy of live or fixed S2 cells, respectively.
Hi, I'm Joey Klebba from the laboratory of Greg Rogers in the Department of Cell Biology and Anatomy at the University of Arizona.
I'm John Nye, also from the Rogers Lab.
And I'm Dan Buster, also in Greg Rogers' lab.
Today we will show you a procedure for preparing Drosophila S2 cells for light microscopy.
We use this procedure in our laboratory to study the regulation of centriole duplication in Drosophila by studying changes in centriole number and structure following elimination or overexpression of important regulatory proteins.
So let's get started.
We use standard 35mm Petri dishes to make our glass bottom dishes. These glass bottom dishes keep cells in a condition very similar to their normal culturing condition, as well as allow live cells to be examined through microscopy-grade glass. For assembly of the glass bottom dishes, a taped surface of a styrofoam box with thick walls is used as a drilling surface. Place the bottom half of a 35mm Petri dish on the drilling surface with the bottom side of the dish against the styrofoam box.